Hello everyone, today I will be presenting my honors presentation. It's called Tracking Provenance of Painted Ladies from Kenya in Winter and in Summer Using Hydrogen Isotopes. My name is Sana and my supervisor is Kama Batay. Insect migration influence ecosystem function and human life. Migratory insects affect ecological interactions by connecting distant ecosystems, transporting nutrients and propagules like pollen over long distances, and structuring food webs. Painted lady butterfly is the most widely distributed lepidopteran worldwide, except in Australia and the Arctic. In order to track butterflies along the migration cycle, H isotopes are commonly used because the composition of individual butterfly wing is expected to mirror the isotopic composition of the local water at the butterfly's natal location. Butterfly wings are composed of keratin, making them a metabolically inert tissue in which isotope ratios are unaffected when an individual migrates. As such, the isotopic ratio of an individual butterfly can be expected to reflect the environment in which they develop as larvae, regardless of where they are found later in life. While it was already demonstrated that the painted lady massively migrates from Europe to the Afrotropics during fall, the existence of a reverse migration from the Afrotropics back to Europe in early spring was confirmed by Talavera et al. In 2018. This research goal is to increase knowledge about migration and spatial temporal origins of individuals across Africa by exploring details of painted lady natal origins from different periods in Kenya. We hypothesize that butterflies collected from November to January will have primarily a natal origin in Europe, while the butterflies collected later in the season in spring will show more Southern African origins. For the methods, butterflies were collected from tropical Africa in winter and summer across 2 years. The sites of collection were chosen opportunistically when hotspots of painted lady abundances appeared on the landscape. A total of 41 samples from Kenya were collected between November 2017 and May 2019. After we washed all wings with a solution of 2,1-chlorophyll-methanol, the samples then ran for exchangeable hydrogen at the John Vaser Stable Isotope Lab. After, the geographic assignments were made using RStudio. We used the growing season D2H isoscape from the assigner package as a basis for this assignment. We calibrated the D2H isoscape in wings using a compiled dataset of known origin insects with D2H analysis in their wings. The isoscape was calibrated by comparing the D2H of this known origin dataset with the precipitation isoscape. The assignments were made using D2H values combined with the species distribution model for precision. To facilitate the visualization of the isotopic assignment, the values were arranged into clusters according to season. If those clusters based on season were not specific enough, they were rearranged according to the U-bird and D index in RStudio. This histogram helps visualize that the values for summer and winter are different. Then, the t-test confirms that the observed difference between both sets of value is statistically different. Therefore, we can say that we have two different painted ladies population, one in winter and one in summer, with different natal origins. Now, we can make geographic assignments with the niche model to help predict the natal origin for each season more specifically. The first assignment is a sum of probabilities of all 18 individuals. We can observe that there is a high probability of the origin being from Europe, but also from Southern Africa. To refine the natal origin, we further divide it into clusters. Those clusters show an important probability of natal origin in Europe, even though some clusters still have a probability of being from Southern African regions. For the 18 samples collected in May 2019, there was considerable variation in hydrogen isotope signature, which implies a broad geographic variation in natal origin. To limit the possible location of origin, we decided to structure this data into clusters. The first cluster is a population that shows a high probability of origin from Southern Africa, but also in Saudi Arabia, Syria, Morocco, or even Spain. The second cluster shows a high probability of origin in tropical Africa, and other regions of origin for this cluster include Namibia and South Africa. The third cluster likely came from Southern Africa or the West African coast. While the geographic assignments remain pretty broad, they appear to support our hypothesis. As per our results, it is probable that there are two population groups who migrate to Kenya, and thus these two populations are separated temporarily. The first group migrates in winter from Europe southward, while the second group migrates in spring from Southwest Africa northward. The population in the southern hemisphere could be migrating to spend the rainy season in tropical Africa. Conversely, the population in the northern hemisphere would spend the winter in tropical Africa and migrate north to exploit temperate biomes during the summer. To conclude, insects are the little things that run the world, and it is crucial to investigate and protect their massive seasonal migration. Thank you for listening.